Hi, I'm Ted Salmon, and I want to talk here about high-resolution video or digitally enhanced DIC microscopy. This is a second talk in a series on DIC microscopy. In a previous lecture, I talked about the principles of DIC microscopy and image contrast generation in the DIC microscopes. Now, a few words about alignment for DIC. To set up the microscope for each objective and condenser situation, you're going to set up for cooler illumination, and usually you start with the low power objective and the specimen, and then work your way up to the higher power objectives, right? And second, you're going to also align the microscope for polarization microscopy, which means in the beginning you're going to take out the objectives, the condenser, the DIC prisms, right? And you're going to make sure that the polarizer is aligned in an east-west direction. There's usually a little double arrow on the polarizer that tells you that the vibration direction for the polarizer. And then you're going to align the analyzer up in a north-south direction. But what you want to do is look down the microscope and rotate the analyzer if you can, and make sure that it makes a maximally dark image. That's called extinction. All right. And usually the manufacturers, particularly today, are really good about this, and they've already pre-aligned all these things, and you slip them in there and you'll get it. But jiggle the analyzer a little bit or what have you, rotate the polarizer a little bit and make sure the two of them are crossed. Because if they're uncrossed at all, that represents noise coming through the system and prevents you from seeing the weakest intensity sort of specimen detail. So the other thing that's really important, just like you would have do for phase contrast, is you have to use the correct objective and condenser DIC prism. So the objectives, there are different schemes that different manufacturers have, but nonetheless they will tell you which prisms should be used for a specific kind of objective and which DIC prism needs to be used to match that prism in the objective. And so much like you have for phase contrast, there's a turret beneath the condenser lens that contains the DIC prisms. And again, the DIC prisms differ depending on whether or not you have what kind of condenser lens you have. If it's a long working distance, low numerical aperture 0.55, that DIC prism will be different than if it's a high NA but dry 0.9 condenser lens, and will be different yet again if it's an oil immersion 1.4 condenser lens. So you can end up with multiple different prisms in order to match the objective prism. All right, and then finally you have to make sure you've got the correct prisms that they're inserted, and then you need to add the correct bias retardation to brighten the image as we discussed previously. And then, and then finally, after you've got the image brightened, then you adjust the compensation for maximum contrast and structural detail of interest, as we discussed. So here's an example image of mismatched prisms. And if you don't have a prism, if you have just one prism, what you see when you look between cross polars and focus on the objective back focal plane is you see a dark line running diagonally in one direction. The prism, the slope of the prism is running this way from essentially 10 to 4, right? And then this is across the prism running from, let's say, what is it, 2 to 8, something like that. And the middle of the dark line is where the thickness of the lower prism is equal to the thickness of the upper prism, so there's no net retardation difference between the two wavefronts at that point. And then as we go off towards the edges of the prism, we get bigger and bigger retardations in the prism, and those are the things that have to be canceled in equally in opposite directions by the corresponding prism. That is, the condenser and the objective prism have to be matched. So this could be just one prism without the other one, or it could be two prisms and they're not matched, and you'll still see this dark bar, right? And you don't get much of a good image. So here we have matched the prisms, right? But we haven't opened up the condenser diaphragm plane in order to match the numerical aperture of the objective. And so we get very poor image quality because we're not getting good oblique illumination that you get from high NA illumination from the condenser. And then here we've got a nice match of both the prisms as well as a fully illuminated objective aperture. And now we get this really nice high-resolution image of my cheek cell. So that's not so hard to do. You have to just methodically work it through. And then often, again, once you have a microscope set up for a certain set of objectives, it's relatively quick to implement and use. Now, in polarization microscopy, you can color an image by using a compensator that's has a retardation that's equal to the wavelength of green light. So 550 nanometers would be the retardation that you would put into an E and an O wave. And if you illuminate the specimen with white light, what happens is the background experiences a 550 nanometer retardation, which for green light brings the E and the O wave, so you're coming along and go thwomp, and they go, one goes faster, one goes slower. But in fact, if you go a full wavelength, that's the same as lining up the two sine waves again, and you end up with the initial plane of polarization, and that's canceled by the analyzer. And so it's called a red plate because when you illuminate the specimen with white light, right, you cancel out green light and let just blue, right, and red light go through, and that produces sort of a magenta background color. And if you have retardations in the specimen that add to the retardation of the red plate, then that will in turn cancel the red light and leave you with green and blue coming through, which is sort of the color of the water in the Caribbean, right? Very light bluish color, right? On the other hand, if you have retardations that end up canceling subtracting from the retardation of the red plate, then you'll end up with a net retardation that's equivalent to the wavelength of blue, and then you get an image that's made from red and green, and that looks yellowish. And so you can see, you know, particularly refractive boundaries in colored red and blue. And I have some images here that sort of represent that. Now, that's how you see, like in NOVA and other places where people have used DIC, and they make these nice colored movies and how they got them to be colored. Now, you can't always be sure anymore because people can do digital processing and do that, but at the early days, that's how they did it. Now, I'd like to say a few words about high-resolution video or digitally enhanced DIC microscopy. Now, if you look down the microscope at this specimen, you can't see the microtubules. The eye is sort of a logarithmic detector and is unable to see the little bit of scattered, the contrast from the little bit of scattered light from these native microtubules relative to the background. But with the awesome power of electronic contrast enhancement, you can bring up this scattered light and make these objects visible. And in order to do high resolution, high contrast imaging, particularly of these very fine structural details, it's important to fully and evenly illuminate the objective aperture, like a 1.4 NA objective. That means oil immersion for both the condenser and the objective, and no air bubbles, right? And it takes practice to learn how to have no air bubbles, right? You have to match the DIC prisms, and you need to have what are called strain-free objectives. That is, in the manufacturing of objectives, if there's stresses or strains in the glass or the cements, this can produce birefringence that makes a noise, and that noise will obliterate the contrast that would be coming from a weakly scattering specimen. Nowadays, we mostly use polarization-type polarizers and that have what are called 35% transmission efficiency, And we use filters that have almost 100% transmission efficiency. And so this is easy to do. You have to accurately cross the polarizer and analyzer, which I mentioned earlier. And that turns out to be very important. A lot of people don't pay attention to that, and the manufacturers are off by just a little bit of where they have the lines marked. And that can make a big difference in the noise in the background as to doing that. And as I mentioned, about 1/15 of a wavelength turns out to be a good bias compensation. If you're going to do high resolution, you have to have vibration isolation. So if you can hit the wall or the floor around you and your image disappears on you or something, then you know that you've got vibration problems and you won't be able to see the highest resolution. All right? You need to correct if possible for spherical aberration, and so if you're working like microtubules close to the cover slip surface, the objectives in oil immersion are corrected for that. If you're going deep into water, then you have to use water immersion to deal with that. Finally, we have always used a magnification to the objective that's 3 times the pixel size divided by the optical resolution. So for example, if your camera pixels have 7 microns pixel size, and your optical resolution determined by the Abbe formula is 210 nanometers, let's say, then the magnification you're going to need to the camera, between the specimen and the camera, is 100 in order to achieve the full maximal resolution capabilities. And finally, if you're going to really be able to define position accuracy well, you're going to need 10,000 or more photons counted per pixel on the average for good signal to noise. Right? So you need bright light sources or long enough exposures. Now, I just made a kind of a plot of the measured fluorescence of a 100 nanometer bead, right? And then the theoretical point spread function, which is this line that's just inside this solid line here. Right? This is the bead size here, and then this superimposed on this is the projected images of the pixels. And so you can see with 3 pixels per radius of the airy disk, you can define very nicely what the airy disk really looks like and be able to resolve to adjacent structures at the theoretical limit of resolution. Now, brightness of the light source depends on magnification, time, and resolution. And you remember that the intensity in the image is given by the 1 over the magnification squared. So the higher the magnification, the lower the intensity is. The illumination that you need also varies with 1 over the exposure time that you have available. So if you're doing things at video rates for video work, right, you need 30 times the intensity from your light source than if you were willing to just do time lapse of something with a 1-second exposure, where you can use a much lower intense light source. Now, video needs a bright light. We, in the past, have used mercury light sources with fiber optic scramblers. Nowadays, you can get quite intense light-emitting diodes that can produce the 10 milliwatts of, let's say, green light that you need to make a nice image of a microtubule, right? On the other hand, if you're doing time-lapsing of things happening in cells at slow rates and integrating for, let's say, 200 milliseconds to a 1-second exposure, then even a quartz halogen lamp is sufficient with using, again, heat reflecting filters and green light filters if you're going to do live cell work, right? Now, it's good to have some resolution tests for what you're looking at, and these are my squamous cheek cells again, looked at with a 20x objective with an NA of 0.45. And then if you use a really well-aligned microscope with the highest resolution, this was a 100x objective, NA 1.4 DIC objective with a fully illuminated objective aperture with a 1.4 NA oil immersion condenser, you can see at the periphery of these cheek cells all these little ridges that are separated by about 1 micron, really nicely defined. So you carry around with you a pretty good resolution test for the performance of your microscope if you can see very nicely the fingerprints on the surface of these cells. The other thing that was or used to be available from Carolina Biological Supply, and I don't know whether they still are, is they sold these Diatom test plates. And Diatoms have a sort of a silica pillbox structure, an upper layer and a lower layer. And the silica shells have genetically encoded regular arrays of pores in their surfaces. And this one here is called plurisigma. And the separation of the pores, which I'm standing on here, right here, D. I guess I have to come over here a little bit, right about there. This is 0.61. And Cirrarella, which is this one here, is about 0.41 between each of these holes. And then this one here is called Amphiplura, this little guy at the very end, right? And it has rows of holes. And the rows are separated by 0.24 microns, or 240 nanometers, and the holes are separated by 170 nanometers. And these images were taken in green 550 nanometers light, and you can't see the holes, even though we're working at maximum resolution. If you put a blue filter in there so you can shorten the wavelength, then the holes become very easily distinguishable. I hope these can still be made available. They're really nice for checking out your microscope. Now, a couple words about video-enhanced contrast. This technology was discovered and advanced by Shinya Inoue and the late Robert Allen in the early 1980s, and rapidly taken advantage of by a lot of neurobiologists, cell biologists, and others to look at using video cameras to look at dynamics of cellular fine structure. And this was a published image from the Inoue lab of how this is the view by eye, and this is the video-enhanced contrast image. And I made an example here of what our microtubules look like if you look by eye, and then if you use the contrast enhancement features of your camera or your computer image processing system, you can bring up the microtubules on the surface, you can see that, but you also bring up all the inhomogeneities in the illumination system. And so what you do is you defocus a bit, and these inhomogeneities don't go out of focus that fast, and you store this as an image, a separate image, and then from then on, you subtract this image from the incoming image, and now you can see very nicely the microtubule sitting on the surface of the glass. And if you do a little frame averaging, sequential frame averaging, you can make microtubules look like telephone poles. So these are very important for video assays, for people who study motor proteins, very important in the life and career of my host Ron Vale, for example, and the discovery of kinesins and other things that they've done. And very important in looking at a lot of the dynamics of cellular fine structure at the limit of resolution. And so this just summarizes this: the view by eye, analog contrast enhancement, or this is a slightly defocused image to store the background image. This is after subtraction of the background, and you can look at live video rates, and this is the image increased in contrast by sequential averaging. So I would like to just finish up by showing you a couple more things. One is this nice video-enhanced DIC time-lapse of a new lung cell. The time is given up here in hours, minutes, and seconds. And we start in prophase. This is the nuclear envelope, so you can see that very nicely. And this is one of the centrioles of the spindle pole that's in focus. The other spindle pole is down here and is out of the plane of focus. And with this contrast enhancement, you can make the depth of focus in the specimen as narrow as about 250 nanometers. And Shinya Inoue has a nice paper on that. So if you search for that, somewhere around 1989, 1990, it's very nicely demonstrated there. And so this is a fairly thin optical section through this nucleus, and these are the condensed chromosome arms, just about ready that get into mitosis at the time when the nuclear envelope breaks down. And so the movie will start here with the entry into mitosis. And you can see great detail about what's going on. Here's this one pole way over here. I don't know how to get there. There it is right there. And the pair of centrioles, sometimes you can see both centrioles. And then you can see individual kinetochore fibers that connect the chromosomes to the spindle. And then finally, we go into anaphase, and here comes the furrow. And so this was used to study in great detail by our lab, actually, the movements of kinetochores and motions in cells, and define this kinetochore directional instability, their ability to move with polymerization and depolymerization of their kinetochore microtubules a number of years ago, because you can make I think we did this by video, and every image was taken every 2 seconds, and so you could get very high temporal as well as spatial resolution. Now, we've also used digitally enhanced DIC microscopy, and this is for budding yeast mitosis, and this was kind of the first set of imaging that was done in order to define the cell cycle time in budding yeast, which is a model genetic organism that's used, in fact, to study many aspects of the molecular and protein functional aspects of how mitosis works. And this is the first time we were able to image the actual 2-micron mitotic spindle that forms intranuclearly in the budding yeast here. And then these subsequent images are the elongation of the spindle as anaphase starts. And then back over here, oops, I got to come back down here. You can see in the next slide, I'll get over here, we progress further into mitosis with further nuclear elongation. And eventually the nucleus puts one set of chromosomes into the bud. And then down here on the bottom, after it's elongated that all out, it then divides the cell into 2. And so DIC was very important in setting up cell cycle timing for yeast. This then was used to look with fluorescent protein labeled cells how different proteins function in the cell cycle. And I want to stop with one last point, which is DIC is often used in a multi-mode setup, like to provide structural images of cells at the same time that fluorescently labeled proteins like GFP labeled proteins tell us where proteins are in cells. And to do that, we need to have a shutter on the illumination path that we control. We can sort of leave all the DIC optics alone here, leave the prism in the light path, and you have to have a system for selecting what the illumination here and the wavelength you want to use for the DIC imaging is going to be the same wavelength that can be passed by one of these filter cubes. We often use a dichroic mirror in the light path that can do multi-wavelengths, blue, red, and green, so that lets you just use excitation filter wheel over here to select which color fluorophore you're going to excite. And then there's an emission, an analyzer, emission filter wheel. And one of those openings in the emission filter wheel contains the analyzer. And you have to put that analyzer in the light path when you take the DIC image. But because that analyzer absorbs more than 50% of the light, first of all, it's going to absorb light vibrating perpendicular to its transmission direction, right? So light's coming this way, you're not going to go through. But even for light vibrating in the direction of its transmission, you still absorb about 15 or 20% of it. And so it makes the fluorescence image very unuseful. And so you have to rotate that out of the light path. And so in the beginning, we did that through rotating the filter wheel. The manufacturers now have rotatable nose pieces, so you can put the analyzer in one of the slots instead of a filter tube, for example. And some of the manufacturers actually have an analyzer that goes in at just the right place, but but what happens is that it has a little motor on it, and it can either push the analyzer in or take the analyzer out, so your computer can control whether it is or isn't in the light path. Right? We get to my last slide. Then you can get these very nice images where you can have DIC of the structure of the cells, like this yeast cell here. And we have actually two fluorescent markers, one for kinetochores bound to chromosomes in this yeast cell, and then a red marker for proteins that are at the spindle poles in the yeast cell. And then this can be either in a time-lapse movie or part of a screen for whether or not you've got normal spindle assembly or what have you. So thank you very much.